Hello, everyone. Just waiting for a few more people to join us, and then we'll get started pretty close to on time here. We're just going to let in a few more stragglers here. All right, so welcome everyone to the Agricultural Genome to Phenome Initiative training workshop using remote sensing to monitor nitrogen stress and yield in alfalfa. So I'm Nicole Scott. I'm the project manager for the AG2PI project. Sorry, and I'm still letting people in, so I'm a little distracted. So this project is funded through USDA NIFA, and this particular training workshop is the result of one of our seed grants to Dr. Biswas and Rios. So I'm going to introduce them really quickly. Dr. Anju Biswas is a postdoctoral fellow participant at USDA Vegetable Lab in Charleston, South Carolina. She recently completed her PhD in agronomy from Forage Breeding and Genomics Lab at the University of Florida, where she specialized in plant breeding utilizing phenomics, genomics, and crop growth modeling. Dr. Esteban Rios is an assistant professor in forage breeding and genetics in the agronomy department at the University of Florida. His specialization in plant breeding and genetics, quantitative genetics, and forage production are the foundation of his research program to improve yield, nutritive value, abiotic, and biotic stress tolerance in forage species. And with that, I will hand it over to Dr. Rios to go ahead and get things started.
All right, thank you, Nicole. Let's see. Can you see my screen on presentation mode?
Yes, thank you.
Perfect. Let me hide these. Good. Okay, so my name is Esteban Rios. I'm an assistant professor in the agronomy department, and today I will be talking to you briefly about alfalfa breeding in Florida and giving you a little bit of a background for our study. And then Anju will jump and perform the demonstration for the image analysis for hyperspectral imagery. So basically, the outline for my presentation, I will spend about 20 minutes talking about current and future efforts in alfalfa breeding in terms of defining the breeding objectives, assembling genetic variation, and developing and selecting progeny in alfalfa. So the livestock industry in Florida, but also in the southeastern United States is very important. We have cow-calf operations for beef cattle, and they normally do grazing. And then we also have a very large dairy industry. And this picture in the center is one of my own farm trials in Central Florida in a dairy farm. Basically, what we are trying to do is introduce alfalfa into forage systems with warm season perennial grasses. The brown color here is basically Bermuda grass that is dormant during the winter. This picture was taken in January. And then green portion is alfalfa growing during the winter month. So the goal here is to basically introduce alfalfa into these forage systems to expand the growing season, and so farmers can have fresh forage year round. We also have some farmers interested in monocultural alfalfa for hay production, particularly for our horse industry in the state. In terms of defining breeding objectives for looking at environmental conditions, these maps represents basically the shift for plant hardiness zones that we will experience in the next 50 or 60 years. So basically the picture in the left is the current plant hardiness zones, and we can see how basically the weather will get hotter, and some of these new plants or new cultivars will have to be adapted to new conditions. Alfalfa has different fall dormancies. Basically, the fall dormancy ratings go from 1 to 11, and the lower the number, the more dormant the cultivars are. So basically, in northern United States and Canada, we plant dormant alfalfa, dormancy groups between 2 and 5. And in the southern United States, we plant non-dormant alfalfa with fall dormancies between 5 and 10. So basically, our program is looking at identifying these regions where non-dormant alfalfa will grow in the future and developing a good germplasm for those future conditions. Alfalfa is known as a queen of forages, is the most important perennial legume in the world. There are 30 million hectares planted globally and about 7 million in the United States. It's the third most valued crop in the United States with about $9 billion per year. It fixes a significant amount of nitrogen and it provides high quality and high yields for forages. In our program, again, we focus on non-dormant alfalfa, and that's basically the base for our breeding program in terms of fall dormancy. We target complex traits, particularly yield, not only the total yield per year, but also the yield distribution, as that is a very important characteristics, as you will see in my presentation. We also look at nutritive value or the quality of the forage, and then some projects on biotic and abiotic stresses. And in terms of abiotic stress, we have a current study looking at nitrogen fertilization to see if we can improve yield, particularly during the summer months. And I'll jump into that in the next few slides. Alfalfa is not really a common crop in Florida, but there has been really good success stories. So this picture is from the 1920s, looking at alfalfa production in the state. And then more recently, with new and adapted cultivars to our conditions. This picture was taken near Tampa, and this is Florida 77, one of the alfalfa cultivars that was released by the University of Florida. But this whole breeding program was dropped in the 90s. And then about 7 to 8 years ago, we restarted the program, and then now we have a new cultivar that was released last year. This is one of our farmers that is testing the cultivar on farm. So we're very excited about developing these new cultivars adapted to our conditions in the southeastern United States. In terms of genetic variation and how we restarted the breeding program, we introduced a lot of germplasm or non-dormant germplasm from different sources. We have about 7 populations of alfalfa that was at UF, but we believe that that's not enough to really have a long-term breeding goal to improve complex traits. So we introduced materials, particularly from Argentina and other sources in South America, as well as other breeding programs from the United States. We grew these plants in Citra, Florida for 2 years. And then this graph represents the dry matter yield across different harvests. And then each line is the average for the germplasm from each origin. So this blue line represents the 7 populations from UF. You can see that in the first spring after planting, we don't see much differences between the germplasm sources. But it's after the first summer, here is June, July, and August, that material adapted to Florida basically maintain better yield during the fall, and then has a spike in yield again in the second spring. While germplasm from other sources, they start dying because of abiotic and biotic stresses, and then they cannot persist for more than 1 year. We selected some of these populations that had higher yield, and we used those plants for crosses to develop our new breeding materials for the future. Just to give you a snapshot of how they look like, this picture was taken last year in some of our plots. You can see here this particular family having really low yield, low amount of leaves, and basically almost no adaptation to our sandy soils. These soils are sandy, has between 70 and 80% sand, very low organic matter, and so basically very low nitrogen in the soil. And these plants being under stress for heat and for some of the biotic stresses, they might also be not fixing a lot of nitrogen, and that's kind of the rationale for applying some nitrogen in alfalfa and see if that can improve persistence, but also increase yield over time. So we selected these better families or better genotypes and developed our new training population. This was done in late 2018. And now we are in the third cycle of selection for alfalfa, but the first cycle was developed by crossing 27 genotypes with high yield from different sources, from Argentina primarily, and also the United States. And we crossed to 7 male parents, 4 of them coming from the UF breeding program, and 2 commercial cultivars that perform well in the southeastern United States. And that's basically how we develop our training populations for the application of genomic selection in the breeding program. So our lab spends a significant amount of time looking at this equation and thinking, how can we improve the efficiency of the breeding program? At the phenotype level, we try to implement new technology to speed up the process of data collection and basically make the program more efficient. We are using genomics in order to make selections more accurately, and then better experimental designs, environmics, and crop growth models to understand genotype by environment interaction. Why are we using high-throughput phenotyping? Well, again, we have alfalfa, a non-dormant alfalfa growing throughout the year, and we basically can harvest these crops every month, so about every 28 days. That brings or that generates a significant amount of labor and costs in the breeding program. So by applying or by implementing high-throughput phenotyping, we can potentially reduce the amount of labor when collecting ground truth data. And again, this graph is real data from our breeding program showing you the cycles for high yield in the spring, and then how yield starts declining in late summer and early fall. Basically, this is part of the AG2PI project that Andrew will be presenting the imagery analysis for hyperspectral drones or hyperspectral sensors. And this is a project also in collaboration with Dr. Aditya Singh. He's an assistant professor in the agricultural and biological engineering, and also his graduate student, Ayush Sharma. So the early work in high-throughput phenotyping was done with multispectral images, and this was part of Andrew's PhD studies in my lab. This graph represents herbage accumulation or yield in alfalfa, and then the y-axis is NDVI. We can see that we have a good correlation between NDVI and yield, but that's really not the question that I'm trying to answer in my breeding program. So my goal is to increase genetic gain for complex traits, and yield is one of them. So in this study, what we did is we have a kind of a simulation looking at the question was how many plots or how much ground truth data do we need in order to maintain genetic gain if we use remote sensing. So this 100 represents that we have ground truth data for every single experimental plot, and we have the remote sensing data. And then we started inserting missing values in our data set up to 30%. So we basically reduced the amount of labor by 70. And then we fitted different models, univariate models for NDVI, for herbage accumulation. And these 2 models, as we increase the amount of ground truth data, the genetic gain for these traits increases. But look at this top line here. This represents a bivariate model when we combine high-throughput phenotyping with ground truth data. And it's basically telling us that we can reduce up to 70% the amount of data collected at the field level and still maintain genetic gain. This is good results, but I'm not focused on maintaining genetic gain. I'm focused on increasing genetic gain. So this is telling us that we have the potential to increase our population sizes more using remote sensing and reducing the amount of labor at the ground level. We are also using NIRS to develop our prediction equations, and also using phenomic selection with the output from NIRS as well. In terms of some more results for our work on genomic selection and some other areas, we applied genomic selection in our program, but we have to modify the methodology a little bit because genomic selection was or is being implemented at the individual level, but we are working with cross-pollinated species. We are running genomic selection in annual ryegrass and alfalfa, And these plants are basically outcrossing, and a single cultivar is basically a representation of multiple genotypes that perform similarly. So what we are doing is creating these training populations that are either full SIP or half SIP families, and then we phenotype them in swords. And the genomic data is basically a bulk sample of DNA from multiple individuals within each family, and then we calculate allele frequencies for each one of the families, and we feed genomic selection models. Some of the early results, looking at only using molecular markers. This is a project that we obtained a little bit over 100,000 markers with rapid genomics. And the models are GBLUB using tenfold cross validations. We can see how predictive ability changes across the year, across multiple harvests. And this is basically a very similar trend with higher predictive abilities in the spring. And then as the plant performance is compromised during late summer and early fall, we also see a decrease in predictive ability. Then the question was, how many harvests do we need in a given year in order to make predictions for the whole year? So we started increasing our training set with increasing data from consecutive harvest. And we see that the predictive ability basically plateaus between 5 and 6 harvests. And that is telling us that we could potentially reduce the amount of harvest by 50% if we implement genomic selection in our breeding program. So we get an average predictive ability for yield around 0.3, which is also in alignment with several other studies in alfalfa. My graduate student, Pablo Sipovics, is testing a new genotyping method developed by Breeding Insight. And the good thing about this project is we are using exactly the same DNA samples that we use for rapid genomics. So we basically tested the same DNA with the two methods. With rapid genomics, we obtain about 114,000 markers. And with Breeding Insight, we have about 2,200 markers. And again, we are using allele frequencies because we are working with populations. The bottom line is that these are really good results. Both methodologies are giving us very similar predictive abilities for yield. In this case, the x-axis is different training sets by incorporating data for multiple harvests in order to predict the total yield in a given year. Again, by having 4 to 5 harvests in the model, we can predict the total yield with a similar statistical significance. And again, overall, the predictive ability between the two methods is around 0.3 for dry matter yield. Now, we see a lot of potential for the new markers developed by Breeding Insight because the cost is significantly lower, and that will allow us to implement genomic selection within our own breeding program in the future. Lastly, my postdoc, my former postdoc, Claudio Fernandez, he implemented envirotyping in our breeding program because, again, we saw this genotype by environment interaction in terms of seasons within the year in our environment. So basically, we use the software EnvirArchetype to create these kernels with environmental data during the period of the study. And then looking at different physiological parameters, we determine whether the plants were growing under stress or under non-stress. So these yellow colors represents harvests when we expect alfalfa to be growing under stress, while the blue colors represents harvest that alfalfa are growing under optimal conditions. In terms of some of the results, we fitted these models for multiple traits, but I'm going to be showing you only yield and persistence for the sake of time. And then the first model here on the left represents the predictive ability in the y-axis. The x-axis is the number of harvests that we include in the prediction model or in the training set. And then this is only GBLUB, basically ignoring any genotype by environment interaction and ignoring the environmental information in our models. As you can see for yield, as you increase the number of harvests in your training set, your predictive ability increases. But for persistence, if we ignore genotype by environment interaction and environmental kernels, we cannot develop good models to predict the persistence of these plots. And persistence basically means these plants will not die and will be growing under multiple years in the future, which is something very important for perennial species. Now, as we start incorporating genotype by environment interaction terms into our models and the kernels for environmental information, predictability for persistence increases. So this is showing us that we can incorporate envirotyping and improve the predictability for these complex traits in alfalfa. Lastly, just to introduce you to the trial that Andrew will be presenting, the pipeline. This is a trial that's part of my PhD student, Pablo Sipoic's PhD program. And he is collaborating with Andrew in this particular study. We have basically two environments. We have zero nitrogen. So basically we don't fertilize these plants ever with nitrogen. And then in the other environment, we apply 30 kilograms of nitrogen per hectare after each harvest. We have 150 hapsip families. They are all non-dormant alfalfa. The trial was planted in March 2021. And for yield data, we have already 7 harvests. And then we perform nutritive value using wet chemistry and NIRS in these 3 harvests. So basically at the end of the peak for yield in June 2022, in August, and then in October 2022, looking at trying to see if we could model not only yield, but also the nutrient content in the tissue of these samples. We extended the work, and we are also looking at fiber and digestibility across the multiple seasons. Angie collected a multispectral and hyperspectral imagery, again, in these 3 harvests. And this is the data that she will show in you after my talk is finished in the next few minutes. We are also genotyping these populations with breeding insight markers in order to develop genomic selection models, integrating phenomics, genomics, and also environmics in the near future. In terms of preliminary results, you can see that the plots or the experiment receiving nitrogen, it produced a significant amount of more dry matter per hectare than plots without nitrogen. But our hypothesis that the nitrogen will increase yield in the summer or early fall basically is not right or is not correct. And that means that there might be other factors, particularly abiotic factors such as heat stress, that could be impacting yield at this time of the year. And that's what we will be looking at in the future with Pablo. He's trying to develop some crop growth models integrated with genomic prediction. And this is the methodology that Mark Cooper and Charlie Messina used in Corteva AgriScience. So we are trying to model some of these parameters similarly in alfalfa with 2 extensive data sets. One is for the United States, and the other, or the second data set, is coming from Argentina, trying to look for adaptation of different fall dormancies to different target environments, and developing new breeding methods for alfalfa in the future. So with that, I would like to thank AG2PI for the funding, particularly for the nitrogen stress study, and the rest of our lab members for their help with data collection and plot maintenance. With that, I will be happy to take any questions.
So if anyone has questions, feel free to just pop them in the chat, or if you're comfortable, I guess you can unmute yourself and go ahead and ask.
Esteban, Jeff Olenek at Purdue. Nice presentation, enjoyed it. So what can you tell me about the soil pH and other attributes in the last part of the study where you're applying nitrogen? Because oftentimes we don't see alfalfa respond to nitrogen, but in Florida we do. And were the non-fertilized plants nodulated properly? Just kind of curious. Thanks.
So the soils are very sandy. I don't know if you notice in the picture, but it's basically very high sand content. I would say between 70 and 80%, very low organic matter. So also in terms of water retention, very low water retention, and so on. So I think that's part of why we see a slight increase in yield. But again, our hypothesis was that we will see that greater response later in the summer when plants perhaps are under more stress and maybe not nodulating and not fixing. We have not done an extensive root phenotyping to look at nodulation, but we normally see a decline in nodulation, especially after August, between August and October. I don't know if I answered your question completely.
No, it's just such a very difficult environment for alfalfa.
Right. So, you know, we now started looking at temperature as well. And again, that's part of Pablo's PhD studies, because there might be, you know, some combination of multiple factors playing a role in the very in the decrease for biomass production during those months.
But as the soils, are they typically acidic and low in calcium? I mean, they're probably low in calcium since they're sandy, and that's required for nodulation.
Right.
So are they also the pH is at 6 or less?
It is closer to 6. Is between 5.7 to 6. Yes. I normally don't, you know, in the breeding program, I try not to use amendments because I would like to, you know, breed and select for material that will be adapted to the conditions that we normally see. I mean, growers are trying to reduce costs and so developing cultivars that are adapted to the conditions without adding extra lime and so on is one of my goals. Now, we are not going to be advocating for nitrogen fertilization. This was just trying to select for plants and study the adaptation to, I guess, nitrogen, and select for plants that will still producing higher yields without the need to, you know, applying nitrogen in these plots.
No, I think it's great because that you're looking at, can we get some fixation and some growth going on this plant, make it better adapted to those sandy, slightly acidic soils, because there's a lot of them around and not everybody has access to lime, particularly when you go overseas to some countries in the developing world. So great. Thank you.
Thanks. All right. I think it's maybe time to transition to Anju's presentation. Anju, do you want to share your screen?
Hello, everyone. Can you see my screen and hear me clearly?
Yes, thanks.
Thank you, everyone. Thank you, my mentor, my PI, Dr. Esteban Rios, for his introduction and kind of overall view in his program and where I was working last 4 years and few months as a student and sometime as a postdoc. So we worked together for this project that's hyperspectral imagery pipeline development, or how we process the images. And today I'm gonna present this as a tutorial because some steps of this presentation will take forever, like few hours, sometimes 10, 12 hours. So to get rid of those kind of distraction or like stacking, I prefer to present as a tutorial. And for this presentation, I would like to introduce us other two folks who contributed a lot. Dr. Aditya Singh, already Dr. Esteban Rios talked about him. He's a professor, assistant professor from UF as well, but different department. And Ayush Sharma is his PhD student. So first of all, like why we chose phenomics in our program and why hyperspectral imagery compared to other sensor, And also LiDAR sensor I'll be using as a part of this presentation. It's because we use some part of data from LiDAR. We flew the drone with both sensor. And how does it work? And overall pipeline to process the images. So the thing is, phenomic is a systematic reproducible measurement of any phenotypic traits. In our case, alfalfa was our organism are like to do phenotype. So there are different sensors. One is ground-based, aerial-based, or satellite-based, and two different type of sensor based on whether we need sunshine or not. So whether when we need sunshine, that's called passive sensor. For example, multispectral or hyperspectral. So we need full sunlight to get the data. But for active one, we need not that because it's itself throwing the laser or the ray and getting the data. So okay, so multispectral and hyperspectral imagery, Dr. Esteban Rios showed my previous result from my PhD project about multispectral. So for multispectral takes only few bands, while the hyperspectral takes several bands. So you can see better resolution, precision, phenotyping. At the same time, subtle difference you can see from there as it's captured the different wavelength as a continuous instead of discrete. For example, multispectral had only 5 bands, and as I mentioned, hyperspectral usually have more bands, like 100 or even more. So that's one reason we use that hyperspectral, and also we didn't find any pipeline available that time when we proposed the project. So that's another reason we chose hyperspectral imagery using in our program or in our project. So I'm not deep diving in LiDAR data processing, but we need a DSM file that's called digital elevation surface model or so. So we'll be using that as well as some sort of GPS correction things. That's why I'll be using some part of LiDAR processing, but I'll not go at the end of LiDAR sensor processing. Mostly I will focus on hyperspectral imagery processing. So how LiDAR works, as I said, it doesn't depend on sunshine. So it just uses laser pulses and measuring the absorbance based on that. So here is our image processing pipeline. There are several steps you can see. One is flying the drone first, then transfer the data into NAS, then different correction. One is geometrical correction, atmospheric correction, and at the end, auto rectification. And after all, after the stage is stitching the images, and based on our need or objective, we generated a spectra or reflectance. And in final, it will be using in statistical analysis. We didn't use that part yet. The statistical analysis is still on progress, so I'm not gonna deep dive in that part. And the materials we used for this overall process, our drone from Dr. Aditya's program, then GPS base station, computer sensors, and NAS platform to store the data. 3 different flights were performed for that specific experiment. And also NV LiDAR digital surface model, as I mentioned, we have to use that. We use that for our hyperspectral stitching the image and other data generation. And mostly we use RACE software, 2 different Dracor and PERS, Dracor for atmospheric correction and PERS for ortho rectification, both for hyperspectral imagery as well as for LiDAR sensor. So fly the drone, step 1. First of all, what we did, we have our drone from a Harris aerial carrier, HE6 hybrid. We had the hyperspectral sensor from Headwall Nano. We had the LiDAR, Applanix APX 15 IMU for the GPS data. We have used the field laptop you see in the middle. So basically what we did, we planned the flight plan mission, and then this is our drone. And here, Dr. Aditya Singh, who is a remote pilot for that drone imagery for 1 flight. And finally, it's flying, and you can see the trimble on the ground as well for GPS data collection. And the second option step is transfer data to NAS. NAS is just a central data point system where the team member can access the data point whenever they need. And here is the overall pipeline for like LiDAR versus hyperspectral imagery, how we process the images for geometric correction, atmospheric correction, or orthorectification. So if you see the left workflow is from raw imagery from hyperspectral sensor, then Headwall software, since it's from Headwall company, so they have some software and we use that. For radiometric correction calibration, we use Drucker software. And some other correction, we use PER software, and later on we use atmospheric correction or orthorectification. And similarly, the LiDAR dataset, we use post-back LiDAR QC and calibrated LiDAR point cloud. And then again, we use PER software for orthorectification and atmospheric corrected image. Okay, so how the file looks like. So when we got the data from the drone to our NAS system, we see different files I just got from different sensors. So here is our file for AG2PI and some other file for example, TXT file, like how many times the drone is capturing images, IMU GPS file, that's like more about GPS data point, LiDAR PCAP, all those are more about GPS data or how the drone sometimes is tilting. So orthorectification, all those data points you will see here. And as I said, like Trimble is also, Trimble is for GPS data collection, that's our own system, but you can use the GPS data from base station as well. So from there, we can see, if you can see the Trimble data, the time points when it started and when it's ended. So Trimble data itself is taking the overall time frame when the drone is sitting on the ground to when it's flying and when it's coming back to the ground. So from ending, starting to ending data point is capturing by APX, it's called Aplonics from LiDAR. Then the Trimble, as I said, that's our own base station. That's called TO2 file. And you can see 1410, that means 2 p.m. and 10 seconds or so. And the last one, you can see 1520, that means 3 p.m., something like that. So it's somehow giving the when the drone was on the ground and when it's ending the flight. So it's giving all those time points. But from IMU GPS file, we can see the drone flight time again. So 15:0031. So this one is the exact time point when the drone was in the air. And the last time you can see the stopping time is 15:0031. So our point is we want to reduce the time when the drone was sitting on the ground and when the drone was coming back. So unnecessary time points we wanted, we did that part for removing that time point. So that is the IMS GPS text file, as I said. And here, what we will do based on that time point, if you remember IMU GPS file, so we have to follow the starting time and ending time from there to get rid of the other time points like the idle sitting time of our drone from Aplonics file. So what we are doing, if you remember, the time point was somehow close to this time point, is 1457. So we are getting a little bit before the flight and a little bit after the flight. So you remember that it was 3 p.m. and few seconds, and we are keeping it 15:12. That means 3 p.m. and few seconds. So we are just copying those and we are reducing the other one. And in our folder, we also had some other files that's called PCAP file. As I mentioned, that's from LiDAR file. That's why we needed the LiDAR sensor data for our hyperspectral imagery eventually. So we have to copy all those PCAP files for another folder. And we created this folder called PCAP because we need that for data processing for geometric correction. And we used post-spec UAV app planning software to process that imagery. So what we did, so we opened that software, and from this, we chose the new project. And from new project, if you see that one by one, I chose that PCAP file, if you remember the last step. And from there, we are taking this app planning folder. And if you remember, I reduced and got the exact time point. That's TO4 data. It's app planning file. It's called TO4 file. So, and Trimble, it's called TO2 file. So we are getting those data points, and it takes time to load those data point importing in the software, but it takes few seconds or few minutes, depends on your computer processor and all. And it just get things done. And you see that there are some noise. It's because maybe our drone was sitting for a few minutes or a few seconds, like it didn't fly that moment. So our target is to reduce those time points. And another thing is we can choose a base station that's available nearby our research station. So you can see that if you choose find base station on top of this bar, if you see there are several options, but you can see find base station if you follow my cursor. So from there, we can see like around 200 kilometer, how many base station we have, but always we choose like the closest one. So the closest one was for our case, Ocala, which was 25.666 kilometer. And here you have to choose the option, that option we choose for none. And since it's a single base station, that's why we chose single base. So that's how we have to import that base station to fix our images. And this process also takes a few minutes to get things done. And you can see that after that, we have to choose there is another option, GNS interior processor. So that part takes another few seconds or few minutes. And after that, you can see that whether our image is fixed or not, based on the base station. If it's fixed, then it's great. If it doesn't fix, definitely we can take advantage from our own base station that's called Trimble, I showed in the beginning. So what we see the green color, you can see the fixed, it's fixed almost 100%, like other solution. When it's 100%, that means we need not to worry about our Trimble data points. We definitely can use. And there are several options we have to choose for this stage. In our case, we use the Ocala base station. So we chose the model, and you will get a message. This called the initial base rover separation for forward or backward processing exit 20 kilometer. Yes, it exit 20 kilometers. So we just choose yes. And it takes little time for that part as well. But what I don't know whether you can see that two different lines. The pink line is our initial line for our drone flight, and the green line just adjacent to the pink line, that is our geo correction part. After using the base station, it corrected the GPS. And if you remember, there were some noises before here, and all things almost gone, and our images is corrected. I just kind of tried to zoom it so that you can see. I'm not sure whether still you can visualize the things clear. I can see that, but maybe you can see that as well. And we have to save this image for further processing, right? And our experiment name is called AG2PI, so we kind of keep the consistency for the naming as well. So here, there are several files. Already I showed you how many files you can get, but based on your need, based on your software, what files you exactly need to run the software or process that a specific part, we have to choose that. So we have to choose this point mission part. And in mission part, you can see there are, the mission part is coming from the importing a base station, corrected image, all those things. So you can see there are two different files already generated. One is called extract, another is called proc. And here we have to choose the output file, and you see that the output file is called export emission output file. And what we have to do, we have to follow this custom smooth BET that called like SBET file. Export file format will be SBET, and it will start the export data, whatever you are giving it to the system. And you see that there is another folder already created from that part that's called export. So there were two different folders initially, that was extract and proc. Now added another one, export one. So you can see from our mission folder, we have two different files generated already in export file that's called SBET export mission file. One is log and another is output file. We will have, we needed that file for further processing. So we are kind of slowly gathering what file we needed and what things need to corrected for the further processing. So here you can see another in our drone we had lever arms and mounting angle. So you have to choose that to see their what was their X, Y, and G. We have to fix this reference IMU mounting angles. So generally what we did 180 for X, for Y 90, and for G minus 90. It's like kind of fixed by the company people, and we did follow that. And the Applanix file, we have to import that folder in this software again. And if you remember, I showed you some PCAP file from LiDAR. So we are kind of talking about the LiDAR file processing, because we will be needing that for our hyperspectral imagery processing. And here, all those PCAP file is importing in the system. And you see that it's loading, it takes time. And now it's done. Again, you can see that lever arms, all those value here. So you have to fix this part, like the bore side angle specifically 5400. And for the second one is minus 16 if you follow my cursor, and TG bore side angle is 5400 again. So after switching all those options, it takes time to loading. And now you see there is another folder created, if you remember. So slowly we are gathering folder here for different files. So we had this 3, and now it added LiDAR RC, that's called quality control of LiDAR thing. So this part is for radiometric calibration using head wall hyperspec 3. So in this process, we will run this file as a batch file. So what we chose here is a radiance batch, basically. Then we had those files that's called raw header file, and we have to choose all those files here for head wall software for radiometric calibration. And the thing is, if you notice that how many wavelengths starting from 399, since it's continuous for hyperspectral, so you see there's continuous bands and so on. And all those things, you can see the digital number .HRD. Then we are trying to get our file from here. So we'll tell this file name that we need those files. So header file, raw header file, and we are opening this. And the suffix here, since we are doing the radiance calibration, the suffix will be _RD. And after that, we have to start the processing. And it takes time to load that data as well to finish the process. And it's still loading. And finally, after that process, you will be seeing, if you do that by yourself, you should see that a raw RD radiance file and raw HDR file. So we'll get more data based on our text frame index file, like how many bands we have and so on. And thus this step works. And now after the radiometric calibration, we use the Dracker software that's from RESE software, and we will be using these two versions. One is called Dracker version 2, another is for PARS version 3.5. So for Dracker part, since Dracker itself doesn't have the radiance part, that's why we did the radiometric calibration using head wall, and we'll be using that data in Dracker for atmospheric correction as well. So for Dracker software, what we'll do, we'll choose the import option, then head wall hypersp, and then we have to make sure we are choosing for file filter only the radiance file we created, so that it can pick all those radiance file from there if you fix that by _RD. And the data will be there. And in this stage, if your system is kind of more protected, you may get this kind of password needing for that. And we will be using creating a folder called Dracker, because we are using the Dracker pipeline right now to get things done. So basically what we will be doing in this step, we will be adding in this folder. So the next step is you have to see which option you have to choose here. So the ground elevation we chose from global digital elevation model. And we will be using a digital surface model because we found both of them are kind of similar and got same results. So here you notice that radiance file again, and its atmospheric correction is going on. And reflectance processor from here, we have to remove this BISC file from this. If you see that in this nano sensor, there is anything at the end of this file, make sure you are removing that part. So what I'm keeping, just raw RD. So the Dracker folder is still here and taking all those data after processing. So you can see there are several options. Basically, we kept those as our default. We didn't change anything. Then here, run the Dracker. So all those part is done right now. Whatever needed, like output folder creation or other files needed for running the Dracker, it's done. I mean, already in the system. Now you run Dracker, and it again takes time to process things. And now you will see there are several files created under this folder we created for output. So you can see BISC file, header file. There are several options. You see that. So make sure we have to remove this after raw, and the number will keep only from header file and BISC file. And for header file, what we will do, we will keep only HDR and raw _0, and in between RD or our reflectance, whatever it is, we have to delete it to for sake of processing the further processing part. So what we did individually, you can take help from Python code. There is a script to run that for if you have many files. We have it. Later on, we will provide in our pipeline when it will be publicly available. So these are all the things you can see what we'll be needing for next processing frame index data point. We need all those raw radiance file, header file, IMU GPS file, SBET file already we created in our mission as a log and input file. And now you can see that the PARS version will be using that part for next part, ortho rectification part. So in this part, as I mentioned, DM and DSM, so it's kind of similar. So in our case, though, we are choosing here a DEM for import and GOT file, but you will see we are we'll be choosing the DSM file, the TIFF one created from Lightroom, and we have to name that a file like output folder. then it will take time to again import the data or get things done. It says that attention DEM values below this deleted will be reset to 0. So just ignore that. You can just press OK. So display DEM. And in this part, we can see some sort of this specific part I borrowed from another experiment, not from Alfalfa. So you can see that this specific part, basically we can see the top of the plants or so. And to visualize is better, to see the more visualization, we have to choose the LIN 2% so that you can see better, visualization here. So you can see almost the complete field here, and the right cursor here, you can kind of zoom the specific part if you want to. Again, you can run this as a batch file using a coding if you want to. And we are using nano directory processor here. And we have to make sure some options here, which one is the right to process this part. So we have to make sure we are choosing hyperspectral applanics. If you remember, we use the APX file that's called applanics file. And also our FOV should be 32. So all those things I need to make sure everything is all right to run this process or run this specific step. And we have to choose the bore site for the further processing, for stitching the image and all. So you can see I am choosing this. This is Dr. Aditya Singh's NAS system we use. So basically, we had this name for like Dr. Singh. Bore site GCS, we chose that part. And after adding all those things, as I mentioned, hyperspectral applanics 32, and the file, bore site file, all those files needed for this part we chose. And now it's time to define the output directory. You see the cursor here, so we have to define the output directory here. And for output directory, since we are using all bands for this images, so the folder name itself is all band images. And we'll go inside there. And now it's time to process the part. And if you see that there is a chart for our ideal alert window, you can see the GPS range based on X, Y, and for DSM range, all those things you can see from here, altitude. And it's a start processing here. It says that the starting parts processing, all those information you will be seeing in your system. Now, after running the parse system in all band, all image, you can see there are several files already created. So the files already created, the parse sector is, the step is done. So now it's time to see whether our processing works or not. So to justify that, we have to see, our image that's called our RGB JPG image from Dracker. It's a kind of raw form. You can see the field view. And the next step is when you are done with your geometric correction or atmospheric correction, ortho rectification. Now it's time to stitch all those images together and, to generate the reflectance or a spectra. So here in we are using NV Lighter software. Then you see the file. We have to choose from open option here and which file you want to see. We want to use this BISC file from here. You see that all band images, we process the image at the end. So there are several files generated, but basically we have to use BSQ file here. And there were two trials Dr. Rios explained in his presentation, that two different experiments. One is nitrogen with nitrogen, and one is without nitrogen. So that's the reason. And here I'm showing how it looks like when it's it looks green, like you can see all those plots, right? So there were 2 reps, for example, I'm showing here the experiment with nitrogen. So there were 2 reps and all those columns and rows, it's visible from there. So what was the next step? So next step was to create shapefiles. So why we create shapefile? Because we specify the plot, like which plot is that? Because when we generate reflectance or a spectra or NDVI, I mean vegetation index, whatever, so that time we have to specify the specific plot information so that later on when we get the data in our Excel spreadsheet or whatever, we know which plot is giving this result so that we can compare for our statistical analysis. So as I said, the 5th step is generating reflectance or spectra. So basically Dracor software, in RESE software, if you remember, we use 2 different packages, Dracor and PARS. For Dracor, we used here radiance calibrated data, and then we can generate from here reflectance data. And PARS one used for orthorectification, and from here we can do extract our spectra. And later on, one of our goals for this project was to see like the wet chemistry as well as compare with our spectral data or sensor data, how it works, how it can detect our plants are stressed or not. So this part we can compare these data points with the wet chemistry we have done already. So that's all about my presentation. I know it's too much to digest. There are lots of information from the beginning to end, but we will be providing this PDF to Nicole so that she can add in their website or other places so that you can get access from there. Anyway, I would like to thank my all project team members, especially forest breeding and genetics lab, all those lab members who helped us for this project from the field to data processing, and definitely Ayush Sharma, who contributed a lot for this pipeline. And of course, our funding source, AG2PI community, who funded for this project. That's why we were able to do that and present this today. And if you have any further question or like queries about this project or pipeline, we have our contact information here. You can reach to us via email. Thank you, everybody. I'll be happy to answer question. Yeah.
So if anyone has questions, feel free to just pop that into chat, or if you're comfortable, go ahead and unmute yourself and you can ask the question. And again, as Anju noted, we will, because we are recording this, we will have a copy of the recording on the AG2PI website on the same page where you registered for this event. And then she'll send us her a PDF of her slides so we can share that as well. And then you can walk back through again all the steps that she just told you about. Not seeing any questions right now, but I have one I'd like to ask real quick. And maybe you said this and I missed it, but the software that you're using, is all of that free, or do you have to have a subscription to it?
It's not free, all of them, especially Dracor and PARS. RESE software, you have to buy that. Yeah.
Angela, you can go ahead.
Hello. Hi, hello. Yeah, thank you for the presentations, you both. Really interesting. I have a couple of questions, one for the Professor Rios and one for you. About alfalfa in the crop models, how do you measure or how do you start the cycles? Or how do you define these key dates that we call in the crop models, that we put key dates when a crop cycle starts to run the model and when is the end? As it's a perennial crop, there is no planting date, I imagine, and there is just one date for a year. So I'm just curious to know about how you set these key dates in the crop models to run one cycle after another, no, in a row. That is the first question. And another one for Anju, thank you very much. Do you use for this LiDAR data sets? Are you using this data sets of LiDAR to identify changes in terms of or morphological changes, for example, or to define some stages, phenological stages during, again, this crop cycle of alfalfa that can determine maybe some or more using more than nitrogen in alfalfa production, something like that. Thank you very much.
Okay, I think I can start. So thanks for your question. So Anju did one of her chapters for the dissertation was to update the DSAT crop model for alfalfa. So there is already one that was developed for fall dormancy 7 in Spain, I believe. So we were trying to update some of the parameters for the conditions here in Florida. So the starting point for us is a planting date, because even though it's a perennial crop, you need to go and plant the crop with seeds. So it is basically the planting date, and then you just continue the modeling throughout the multiple years. So you will have like the curves that I show you with consecutive harvest throughout 2 or 3 years, depending on how much data you collect. So I guess hopefully that answers your question. But even though it's a perennial crop, there is a starting point that is planting time.
Yeah, thank you. I was just curious about to know if there are small cycles during the year. Yes, we have the planting day for perennial crops, but I understand that there is a harvest, and after that harvest, I imagine that they start another vegetative cycle and so on. So I just wanted to know if you point that those dates in the model. But yeah.
Yeah, so you will have each individual harvest cycle. So in a perennial crop, you go and cut, and then you get a new cycle of regrowth and so on. So you are trying to model each one of the cycles, which is basically each one of the harvest periods, which will be between 28 to 35 days.
Okay, thank you.
And I guess, Anju, you have to answer the second question.
Can you please repeat your second part again?
Sorry, I just wanted to know about if are you using this data set from LiDAR sensors to identify morphological changes or to identify, phenological stages, timing between one stage and another one, or something like that?
Yeah, first of all, thank you for your question. So basically, that was not our goal to use LiDAR sensor, but the sensor was already there. That's why we utilized the platform to get the GPS data or DSM file, because LiDAR sensor give you a digital elevation model from there. Specifically, yeah, you are right. We can check the height, plant height, like their growth stages. We can run like every week or every 2 weeks to see their growth. or height, whatever we need. But thing is, for our case, we didn't do that, but eventually it can be a future direction. Yeah. Is that answer your question or?
Yeah, thank you. I
Mhm.
was just curious to know about how are you using the data from LiDAR. Thank you very much.
Mhm.
And then there are a number of questions in chat, and I see that you're answering some of them in there, Anju. But maybe we can just, I'll read a couple of them and then you guys can answer. So Mohammed asked if using multi-spectral sensor is able to measure the nitrogen stress level, and Anju, you said yes, it is able to. There was a follow-up question, have you done from Sushrita, have you done any quality check on raw data of multispectral imagery? Do you want to maybe just answer that verbally, Anju? I think you answered it in chat, but
Yeah.
just for
So
those watching
yes,
at home.
yeah, I mean, during my PhD, I did the one project completely for multispectral. So for this project as well, though it was not part of our project, it's still I flew the drone and processed the data. So definitely, yeah, we did the quality check as well using AZSoft and QGIS. AZSoft is a paid software and QGIS is a freely available software. So that part is very easy and straightforward to process and quality check of our data. But hyperspectral part is pretty complex. That's why that was my goal to work on that pipeline. Yeah.
Thank you. So another question, what is the depth the sensor can detect?
Depth mean we sense, as I mentioned, it has several hundred or more than 100 bands. So basically, it just takes subtle difference, like very tiny difference it can detect based on resolution, based on all those bands compared to a multispectral sensor. Yeah, so you can go very deep if you want to. And based on your objective, what you want to do, it's a big data from hyperspectral sensor, so you can play with that based on your requirement for sure.
Okay, great. Another question here from Katie Singh. How did you manage or correct the interference of Headwall's hyperspectral system? GPU, IMU antenna with a big Harris drone. Because there should be a lot of interference in image alignment and quality. Is that something that you experienced?
So to be honest, I didn't fly the drone. The drone was flew by either Ayush or Dr. Aditya, but since I involved with the process and pipeline thing, so basically that's why you saw the more pipeline we used for GPS correction or IMU GPS file some steps initially in my presentation as a tutorial. So we had to correct those, like if any tilting is there for the drone or like whatever is happening, we had to use some software for correcting those. Yeah, some values we have to input. But the best guess or like the best person would be Dr. Aditya Ayush to answer this. But from my knowledge, it's like that.
Anju, Ayush is there in the chat and he replied,
Yeah.
our case, we measure the X, Y, and Z angles of antenna and headwall camera,
Yeah.
which further added to the postpack for correcting IMU and exported to SBET.
Yeah.
So thank you, Ayush, for answering.
As I said, he's the best guest. So yeah, he's here. Thank you, Ayush.
I think there is a question maybe for me in terms of the minimum ground truth data. I don't know if this relates to the results that I showed. I haven't seen many papers, but based on my contacts with other alfalfa breeders, they did something similar. So I would say at least, you would need 50%, depending on the quality of your remote sensing and the quality of your phenotypic records. You could go lower than that. Thinking about genetic gain, not thinking about just making predictions. But I don't think there is a ton of data, at least in forages. I haven't seen anything on wheat or corn. Perhaps in those crops, there might be some other studies showing how much ground truth data you need in order to maintain or increase genetic gain. And then there is another question: can this be applicable to animal breeding? I'm not sure exactly what, but I suppose you are thinking about high-throughput phenotyping. I think yes. I'm not very familiar with animal breeding, but depending on which trait you're looking for, you know, I would imagine, for example, for grazing animals or things like that, where you have them in large areas and maybe if your pasture, for example, have different components with legumes or grasses and so on, and you would like to monitor where the animals prefer to go and eat, perhaps that's one area where high-throughput phenotyping and remote sensing can help with that. But in terms of the animal traits, I'm not sure how to respond to that.
And then I think there was a question, and I'm not sure I understand the question, whether your algorithm can separate between nitrogen kind of the variety of alfalfa. I don't know if Abdal, if you're on, if you want to clarify.
What is the question?
The question is, your algorithm can separate between nitrogen kind of variety of the Medicago.
I don't know. I will have to think about exactly the question, but I would, thinking about the variety, I don't think the images can tell you exactly which variety. It could tell you, for example, the fall dormancy of the variety, because you could extract height. So fall dormancy is a trait that is measured based on plant height. So non-dormant varieties are taller than less dormant varieties. So you could potentially at least look at fall dormancies. But then looking at variety itself, I'm not sure it can separate specific varieties. And then in terms of nitrogen, yes, that's part of what Anju and Ajush are working on, is trying to detect nitrogen stress between the varieties, but we don't have all the results yet. But in terms of fertilized and non-fertilized, I believe we will be able to do that. But which variety is being grown, I don't think we can do that yet. Anju, there's a question about vegetation indices or deep learning.
We are using reflectance and spectral data here for nitrogen stress detection. And Ayush is here. Ayush, do you think there is anything else we need to do?
Yes. So can you hear me?
Yeah, we can.
Okay. So we are planning to extract the whole spectra of 272 bands from the head wall sensor, and actually running the partial least square regression models, so we can deal with the covariance autocorrelation problem with the spectral reflectance. But that's our best approach, and also most of the researcher who work with the plant chemistries at some level, they use PLSR as the best probability factor or the component analysis to identify which spectrum bands are working or predicting more of nitrogen or any other nutrients.
Thank you, Aish.
Are there any additional questions? Again, you can feel free to type it in chat, or if you're comfortable, you can just unmute yourself and ask it. So we do have a new question in chat. What approach was used in multispectral imagery for nitrogen estimation?
So for multispectral imagery part, we didn't do that yet. I mean, we didn't do that part yet, but it's not part of our project. But willingly, I just processed the images for biomass estimation, because another part in our project is biomass estimation using hyperspectral sensor and multispectral. So in short, I didn't use the multispectral imagery for nitrogen estimation.
Still have people joining. So I want to give it maybe another minute or two in case there are any final questions. But it looks like we are very likely wrapping up here a bit early. Let's give it maybe one more minute. Any final questions? Again, for those who are maybe just joining, or if you're rejoining us, a recording of this will be available later today. So in a few hours, Anju will be giving us PDF copy of her slides so that you can walk through along with the recording what she explained. And again, any sort of final questions before we give you back about 30 minutes. I'm not seeing anything in chat. And sort of last chance if you'd like to unmute yourself. Don't want to take away anyone's opportunity to ask a question. But I believe, Anju, you had email addresses at the end of your slide presentation as well, correct? So if people have
Yeah.
questions, they can find those. And-
Yeah, I added all those emails for our team members who work for this pipeline and project. Yeah. So.
Great.
Yep.
All right, well, seeing no new questions. Thank you everyone for attending today. Thank you, Dr. Rios and Dr. Piswas for sharing your work with us. Again, this was part of an AG2PI seed grant. AG2PI itself is funded through USDA NEFA. So we appreciate you sharing your work. We appreciate you doing this work. And I guess, you know, everyone enjoy your Tuesday. For some of us, it's the beginning of our summer break. So everyone enjoy hopefully some great weather out there.
Thank you.
Take care.
Thank you,
Bye.
everyone. Bye.